when you will click on all databases at NCBI, there is a option genome, and there is another option SRA, sequence read archive. Whenever you want to download data regarding some genomes or transcriptome, you should select that part, because previously we were downloading data related to proteins. So we have selected the proteins. Now when we are doing work on NGS and we have to select the publicly available data, then we should select genome and SRA, that is sequence read archives. We should select data from that part. Now, what is NGS? Maybe you are aware of these NGS. NGS is basically next generation sequencing. Means by means of sequencing methods, we are trying to find what is the issue in any gene or any protein due to which a disease which is occurring in a particular place, in a particular environmental conditions, or in a particular biotic or abiotic factors. Means whenever we are trying to do next generation sequencing analysis, first of all the question is why? What is the importance of next generation sequencing? Why we are here to do NGS analysis? First question is from where that data will come, because in a particular condition maybe there are some loopholes, maybe there are some issues regarding these diseases are normal plants or animals. So our first thing is we have to select the data very carefully. But if we are going to generate data regarding NGS, then what should be the plan for NGS? For example, I'll I'll show you one thing. I'll give you an idea how to select, how to generate an experiment for next generation sequencing. I have observed in some particular environmental conditions, there are several plants which are suffering from some sort of fungus, some sort of bacteria. In the same locality, there are some other plants which are resistant to those diseases, those microorganisms. The breed is same. I'm talking about tea plant, Camellia sinensis. In Palampur, I have observed that in the same variety, T. Camellia sinensis, there is a plant which is disease resistance and other plant which is disease susceptible. Now our idea is clear. We have to see what are the genes which are responsible for disease resistance and what are the proteins which are responsible for which they have some effect and due to which there is a disease in disease plants. This is the, then what will we do? We will do some sampling of diseased and susceptible plant, and what will we do? We will do some sort of sequencing, mRNA sequencing, and after that we'll compare those sequences. Then we'll compare those sequences. Whenever we'll compare those sequences, then we'll be able to find in which place where is the issue and where is the problem, and we have to solve the problem by some methods. Means what kind of genes are there, which protein product is not expressing, what is the expression analysis of that gene, and how we have to enhance or regulate the particular gene expression for that particular condition. Okay, so so that is our main idea. But in case of humans, when we are going, when we have to do some analysis for a diseased or a a, a normal human, then we have to compare the sequences of a diseased and a normal human. That is why this era is going towards personalized medicines. Means what kind of genes for a particular person in a particular region are expressing or not expressing, we have to find that particular thing. Okay, this is just an overview, means how to generate or how to set up the data, how to find the data. We can find the data from genome, ncbi genome. If data is not available, then we have to generate the data by using some methods like pack biosystem, illumina. We will generate the data, will generate the data by means of wet lab. But after that, when data is came or from ncbi or from wet lab, the main thing is how to analyze that data, because the sequence will be only in the form of atgc. What is the fun, what is that data, how to extract the crucial information from that particular data related to disease or related to some susceptible varieties in plants or a disease or normal conditions of a human. Now if you have any doubt that I have spoken till, please ask me so that I'll clear the things.
Yeah, just need little more clarity on how, like, when you say expression, gene expression, right? What can you tell some examples of gene expression?
No. Gene expression is very late in the eighth step of NGS, means that we have to analyze the NGS in order to find the expression analysis of genes. That is the overview, means why we are doing going to do NGS. How to do expression analysis, that is the separate thing. But we are doing NGS for expression analysis, that is a separate thing. Because how
No,
to
no,
do that, we'll do in our some next lectures, that how will we do this expression analysis. Basically expression analysis, I'll tell you one thing. Expression, what is expression analysis?
what is expression actually? First, that is what my question. It's not.
Okay, okay,
I'm
okay,
not going in detail, but you meant some genes will get expressed and some genes will not get expressed.
okay,
Something
okay,
you said in that diseased and non-diseased one, right? So
okay, okay,
I just
got
want to know what is that
you. Okay,
expressed means. What is
whenever there is a expression of gene, what will it do? It will create a protein, it will generate a protein that will express, and it will shown in our phenotypic level. Whenever we are going to extract the sample, we know maybe there is some gene is not expressing. What is the main thing? Which part of DNA, which part of DNA will see whether this gene is expressing or not? You know, from DNA to mRNA to cDNA. Basically, what will we do? In the process of library preparation, we'll isolate DNA, then we'll make some we'll isolate mRNA, then we'll convert it into cDNA, then we'll go for sequencing. The genes which are more expressing, there are more copies of those genes in a particular sequencing. Means if gene is expressing only one time, there is one copy. If gene is expressing two times, for again for that product, there are two copies. If gene is expressing again, third copy. If gene is expressing again, fourth copy. How many times that gene is expressing in normal and the same gene is expressing in the disease, we have to analyze that thing. If there are four copies in normal and there are two copies in disease, it means in normal condition genes is two times expressing, means 2 by 4 is 4 by 2 is 2. Then we have to divide. 4 times normal gene is expressing, 2 times disease gene is expressing. That's why there is a probable condition of a particular disease. Basically the expression is how many times a particular mRNA is sequenced in a particular experiment. When you will see the data, you will easily find in the first step, same gene is expressing. In the second step, same gene is expressing. Further in another step, same gene is expressing. How many reads of a particular genes are expressing in the entire experiment? That is the thing. Okay. Now, anything else? How will you find that? We'll do some analysis at the blast level, at some functional annotation level, and maybe we'll find we have got, just assume we have got 10,000 sequences of nucleotide sequences. Out of 10,000 nucleotide sequences, 50 sequences are related to a particular single gene in a disease condition, and the same thing is happening in a disease condition, but there are 25 sequences of a particular gene. Understand? Means when you will isolate your data, extract your data, will go for the functional analysis. For example, you have done functional analysis of a hypothetical proteins. Okay, you have some 200 proteins, I think. Out of these 200 proteins, maybe there are 10 proteins which have same function. They are in the same category. That means a gene which is expressing and generated the proteins of the same class, how many times it was expressed? If we have found 5 proteins, 5 sequences of a same protein, then it means the gene was expressed 5 times. That's why in every another cycle there is a protein. But if in the same thing in the normal or disease condition, the gene expression of gene is less, means that particular gene is responsible. We have to cross check those things. How many genes are expressing in normal condition? How many genes are expressing in disease conditions? And how many, which gene is more expressing and which gene is less expressing? And the gene which is expressing in normal or disease condition, whether that gene is related to a defense mechanism or not.
So it means that a gene.
First of all, we have to find the function of a particular sequence. When we found the function, then we have to think whether out, because whenever you will do next generation sequencing, there may be 50,000 sequences, 1 lakh sequences, 10,000 sequences, 5,000 sequences. The main thing is, first of all, you should annotate those sequences, means try to find the function from those sequences. And whenever you will be able to find the function, then see which genes are in the same category, means there is a clustering. Cluster the genes and name, for example, 50 genes are in leucine rich repeat categories, 50 genes are in Werke category. These are basically transcription factors. 50 genes are related to enzymes, 50 genes are related to some transposons. So we have to cluster those things. Now we'll see which gene is related to disease, means which is gene is basically have important role in defense mechanism. And now our next thing is this gene which we have found in our data which is related to defense, because I'm talking about defense right now. If we have to select that particular gene. Now our next step is whether this gene is same in normal or disease condition. If the expression is same, then there is no role of that particular gene for the disease condition. Then we have to find some another gene. Then means we have to repeat these things. First of all, try to identify the genes, and in the next step, try to find which gene is expressing low in disease condition or expressing very high in disease condition. We have to extract that information from a particular experiment. That is basically how to design the experiment. Same thing can be done for if we are trying to improve the quality of our data, quality of our products. For example, a transgenic plant has been generated. a particular gene which is responsible for the growth of a plant in stress condition. For example, in Rajasthan there is a desert. If I want to grow the apple plant in desert, what I have to do? Means this is an idea. What I have to do? I have to find a particular gene which is available in desert plants. I'll try to put that gene in the apple plant and try to find whether this gene is really expressed in apple plant or not, for which I need that apple plant to grow in desert region. Because apple plant is suffering from high temperature conditions, I have introduced the gene which can make the apple plant resistant to that particular environmental conditions. Means this is the idea. Many scientists are working in that area as well in order to improve their crops. Now another example, you may see how many varieties of wheats, mangoes, fruits already available in the market. How the apple previously was grown in the upper regions only. Now, slowly, the fruits are basically migrating towards the lower regions. How? There is some transgenics, there are some hybrid varieties as well, so that by means of those things, the gene which is responsible for the particular survival of a plant or any organism in a particular condition, we have introduced that gene in a particular plant. Now that plant will survive in the that condition as well. That is regarding plants. But for animals, you may be surprised to know that the deleterious mutations in island population is high. Why? Because in island, for example, we can take the example of Andaman Island. How many people they are only stuck to that particular island? How many, which means what is the ratio of population which is permanent living in Andaman Nicobar Islands? Very less. There is a inbreeding. The same genes are transferring. The same gene is transferring from first generation to second to third. The mutation is, because the mutation in one, mutation in second in children, there are 90% chances of mutation. If the mutation is recessive, maybe children will not be able to survive from parents if both parents are recessive. There is some call, sorry for that. If the both parents have some recessive mutation, if both recessive mutations will combine and it will express in child, what will be the consequences of that generation? So the risk of basically, we can say, the risk of extinction are highest in island populations, and that's why there are several islands. For example, we can say the example of in Europeans as well, in Asians as well, there are some separate ethnicities. All these ethnic group, they are just stuck in a particular condition. If they will come, they have to struggle for another place. There is a Partha, you belong from which place?
Chennai.
Chennai. What are the environmental conditions in Chennai?
It's mostly humid.
Mostly humid. And what about some summers or winters?
Uh,
Means what is the
Winters are
average
not.
temperature for summers and what is the average temperature for winters?
Summers, I think it will be around 38 to 40 average, but
38
winters
to
is,
40.
yeah, winters
Okay,
is
okay.
around
But
28, 30.
means that is a normal conditions for human being. But if you come to Srinagar, then what will happen to you? Basically, this is not the topic. The main thing is, when you will come, there may be some physiological changes, maybe there may be some other changes, but if you are continuously here, maybe there is some gene which will be expressed and your body metabolism will try to adopt those particular conditions. That is a part of adaptation as well, but how you have adopted? Maybe your genes are interacting with particular environmental conditions, and that particular gene is expressing, and that gene is responsible to survival for you in a particular cold stress condition. Because if we'll sequence for a person which is living in a cold area and other person which is living in some humid or some in our southern regions of our India as well, either in Sri Lanka, there is a huge difference of gene expressions with the particular environmental conditions. The pair gene pool, it's very difficult to find the pair gene pool, but there are admixtures because population is migrating from one place to another. That's why there is a admixture of population. But you can easily find how these admixture has been taken place, and how, what is the basically evolutionary tree of the particular organism, from where these gene came, because you are aware that the cold stress genes are expressing in cold regions only, heat stress genes are expressing in Rajasthan or other desert places as well, for example hot deserts, not in cold deserts. In, if you are talking about some Leh, Ladakh or Assam regions, the genes are totally different. That's why we often say there is a huge diversity. That's the main problem. If we are talking about drugs or some paracetamol, simple things, for every person in India, we are talking about he is suffering from fever, what will we provide to him? Simple thing, that is paracetamol. What is the composition of that paracetamol? 100 mg. Is it possible for every person 100 mg or 50 mg is required? Why not 1 mg? Why not 10 mg? Why there is no classes of in each category? Some persons, they have taken one tablet and they are okay. Some other, he is lying on bed for 10 days but he is not fine. That means the particular genes are interacting for a particular organism. That's why the human genome research has shifted our research towards the personalized medicines. Personal genome, which gene is expressing and which gene is interacting, how much quantity of a particular drug is required for the particular body in a particular condition, and what are the main steps in NGS are. First of all, we have to design the experiment and we have to assemble the data from that experiment after filtering that data. Means filtering means when we are going for a sequencing method, there are a lot of human errors, there may be a lot of some technical errors. We'll delete those reads which are really not expressing by maintaining the coverage. Because what will we do? We'll sequence the same gene 50 times, so that if there are 2 or 3 times there is a error, then we'll eliminate those 2 or 3 times, and 49, 50, 48 up to 40 will select that this is the most accurate sequence among 50, and we'll pick the single one and we'll assemble that data, and there is a consensus of that data. After that, there are some sequences after assembly. Those sequences will come in 10,000 or 20,000 in, they will be in 10 or 20,000 or maybe 1 lakh in number. After that, we have to go some functional annotation by using BLAST. If you are working on humans or animals, we will select some ideal organism related to humans or some particular animals. If we are working on plants, we have to select some model organism of a particular plant. For example, in monocots or dicots, in monocots we'll select model organism bamboo, and in dicots maybe it's Arabidopsis. In humans, human genome sequence is available, we can select human very easily. For some other organism, we have to see the data which is available, for which we can see the probable function of a particular gene is this. After functional annotation, then what kind of gene you required. You have to select that gene, then see what is the pathway this gene is controlling, what is the annotation, means this gene is working at molecular level, biological level, or cellular components. Then we will see what is the expression analysis of that gene. After expression analysis, we can easily mine the markers from that gene. The markers related to, means maybe there is some SNP, that is why that gene is not working. Maybe there are some simple sequence repeats, that's why that gene is not working. Maybe there are some intron length polymorphisms due to which gene is not expressing. We can easily mine those markers. And finally, what will we do? We'll design the primers of that particular gene in order to validate that particular gene in a wet lab, so that our analysis is true, that we have done by using computational means. If we are successful in validating the data, if we are successful in validating the data that we have analyzed, our experiment is successful. Related to this, I have published at least 5 papers in Nature, scientific reports, Nature. You can easily see by typing my name, Gagandeep Singh. I have done work on Tulsi, I have done work on Trillium govenium, I have done work on tea as well. You can easily find some plants, and you can see there is a workflow chart in order to extract the data, generate the data, assemble the data, differential expression of the data, functional annotation of the data, pathways of the data, finally validation of the data related to some stress condition, related to some improvement of the plant, or improvement of the particular product from a plant. I have also published this work in apple plant as well. You can easily Google that thing. But this is the introduction that I am trying to convey. How to do all these things will perform in our next class. Till now, if you have any doubt, you can please ask me.
That NCBI SRA you were talking about, right? So SRA,
Yeah, NCBI,
archive,
SRA,
yeah.
yeah,
So
that is
that,
sequence read archive.
so
Yeah.
all the
Now you can
sequence
type,
reads,
you
yeah.
can type, for example, human.
Maybe Partha can share his screen, no? And do
Partha,
what
please
you are
share
like
your
saying.
screen
Yeah.
so that I'll suggest you.
You can see my screen.
Partha, your screen is visible. Yes.
Mm, so so this is the SRA you're talking about.
Yes, this is SRA. 1 minute,
So
please. Let's select SRA. Don't type human, just type T. Either Camellia sinensis or T. Now search. See, there is a Illumina sequencing of T. There is a Illumina, what, XYG. Select, click on, there is a Camellia sinensis on your right side. Click on number 2653. Yes, please click on number. Now, see, this is the data which is submitted in the form of some bio projects that they have done. Click on any random 1, 2, 3. Yeah. This is a pair-end sequencing. Now see, RNA-seq for a Japanese albino tea cultivator, they have sequenced this. For now, you can click on there is a study. Here is a show abstract. Why they have done this work? See, they have done some this by bud treatment, bud mutation and low light treatment. They maybe they have some idea related to this. That's why they have tried to find some mutations, and they have given some light treatment for a particular T, and they have tried which genes are expressing or which genes are not expressing in particular conditions they have selected. Now, how you can select the data? Just click on all runs.
Where is
All
all?
runs. There is a sample. In sample, yeah,
Here there is only one run.
oh, no, there is not one run. Just yes, please copy this run.
This one.
Yes,
You want me to click this one?
yes, please click on this. Do you have copied that? I have told you to copy that particular run.
You mean to say this number? This
No
number.
problem. Just copy that run. Just click back and copy that run. Just copy. Copy that run. DRR243745. Just copy this. Not link address. Just copy the whole.
Text is it? Okay.
Yes. Copy. Okay, now click on this only. There is the option of download. Up. Browse, search and download. Above. Yes, there is a. Just click on fast or fast queue. Now paste your run here. Try to find show runs. Does not exist. Now go back. Again back. Click on experiment. Back. Again back. There is the option of all experiments as well. Just click on all experiments. What will we get?
Same page is coming.
No
This is the
problem.
run.
Yes, click, copy this.
This is what we copied.
No problem. Just try to. Click on this.
Faster.
Yeah, faster.
So this.
Yeah, show runs. Not coming. Again back. No problem. There are some problems. I have already just click the copy this experiment DRX 23345. Yes.
This is
Copy this. No, paste. Show runs. Yeah, see this run we have tried to find. See how many reads are there, 26.6 million, and the size of this data is 5.4 GB. Now what we have to do, in the right side there is a fast queue, filtered and fast queue. Click on filtered and then click on fast queue. Filtered and fast queue. Yeah, this is a faster, just click on fast queue. Yes. Now what will we do? Download, and data will be downloaded. And how
Why
much
we
data
are
is?
doing FASTQ here?
Because whenever you are going to do next generation sequencing, whenever you are going to do some next generation sequencing for FASTQ, there are 4 lines, but for FASTA, that is only greater than sign and some first line. For 4 lines, you can easily find how many reads are generated, and the other information related, other information for further analysis in SAM or BAM files, you can easily find. Means this FASTQ files can easily be readable, what kind of adapters or what kind of particular conditions has been applied. We can easily read these files. Whether you can download in FASTA format as well for assembly, but in FASTQ format, it is more easy to analysis according to me. Now you can download, but don't download. Now you can download a single run, but don't try to download because data is 5.4 GB, and you know how to store this data.
What are these options? Like what is filtered and what is clipped?
What is filtered and what is clipped? Okay, filtered is basically when you are trying to download the data, there may be some errors. It will automatically filter those errors, and clipped means the error will remain there. Means maybe there are some adapters. Filter data is again we'll do some filtering after downloading this data. But before downloading the data, try to avoid the errors. Means try to download the best form of the data, excluding all errors. After that, again we will filter our reads in NGS because we need the accurate data. And how much it is accurate? The data will never be 100% accurate. So we'll try to find the accuracy up to 70%. That's why, but this is the data that we are downloading. This data we have to download, but if data is not available, then what will we do? We'll go for wet lab analysis and sequencing. Then the same data in the form of FASTQ, they will provide us. Companies, there are several companies which are outsourcing the next generation sequencing. They will charge 10 lakhs, 20 lakhs, and 2 lakhs per sample. And what will they do? They will generate data in FASTQ format. They will filter some errors, and they will provide your data. So this is the data from Illumina. Now analyze this data. The analysis part will be done by some bioinformaticians because there are several researchers from various universities. What they have done, they have outsourcing their data, and they have given their data to me in FASTQ format so that I'll analyze that data and I'll try to find the gene based on their own idea. For example, a director from Ranchi, I don't remember, maybe Institute of some biologicals, he was working on wheat. He has try to extend the size of the wheat grain. He has given me a data related to that wheat, and he asked me that if there is any gene which has important role in the basically that size of the grain or size to reduce the leaf and try to increase the size of a grain, we should try that gene. So I have analyzed, but maybe I don't remember. I have found some genes, but I have given and analyzed data to him. There are these genes, so please read about these genes, and maybe the gene of your interest may be there. I don't know. I have analyzed and given back to them. But the thing that I have shown you, you are not generating right now. Right now you are just trying to downloading the data which has been submitted by some other people, some other researchers. What was their target? Their target for this particular that you have selected, they are trying to find some mutations. But what is our target? Whether this data is beneficial for us or not, because we have not yet decided any type of analysis. So maybe this is not beneficial for us. So what is our idea for a particular organism? But will we select? We'll select if you are working on some microorganism, type that microorganism, try to find the contrast organisms. Means what is your idea? Based on that idea, you will do this next generation sequencing. Finally, what is the end product? The end product is basically whenever you have found the gene, obviously a gene will be translated into proteins, and the protein has a particular function, particular structure. When you find the structure of a particular protein, what is the structure of that protein, what is the function of that protein, and what is the active site of that protein, what is the docking analysis of that protein, what kind of drug can be designed for that protein. You can do lot of things that you have learned earlier. This is basically the overview of NGS. Now how we will do the NGS? The first step is that you have done to extract the data from NCBI if data is not generated by our own. This step is known as data mining.
So for this particular archive of sequences, they have already got the uh, sequence, is the overall genome.
Yes, yes,
How will
yes,
we know?
yes. Yeah,
Is it like
they have downloaded the data, they have submitted the data. Maybe there is some publication regarding this data as well. Maybe they have some publication. They have, because just try to open one paper, Camellia sinensis Gagandeep Singh, type on Google Scholar. Either Camellia sinensis, either maybe some other Trinium govinium. No, 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 not this, not this. Camellia sinensis. Before Gagandeep, write Kuldeep, Kuldip. Kuldeep.
yeah?
No, 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 no. Again, write another keyword, transcriptome. Basically, we have done work on transcriptome. The first one. Now see what we have done. Just directly go on methodology section. I think this was the methodology section before that. Yes, yes, yes.
Yeah.
But where methodology has been written, please? This is the result, no?
I think no methodology. I think in the results itself, methodology.
No, 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 no. I have written methodology myself separately.
No, no, maybe in the end you can see.
I have written methodology. Methods.
Oh. I think it will be down. Product methods is cool.
Yeah.
Now see how we have designed our experiment. We have done some, for example, I'll see plant material. How we have selected plant material? One resistant and other susceptible response against a particular microorganism, that is E. vexans, which is responsible for blister blight disease. And we have isolated the two samples. One is disease susceptible plant and other is disease resistant plant. And what we have done? We have done sequencing of this plant, which is in next step: RNA extraction, cDNA library preparation, and transcriptome sequencing. After transcriptomic sequencing, what we got? We got data in the format of FASTA. Just scroll.
Well, actually, so you
This
have
is the about
you
plant. This is about plant. Means there are
okay, so
some separate conditions.
you did this in wet lab.
Yeah, I have done this in wet lab. But this is about plant. That plant was 3 years old in these conditions. 7th day, 14th day, 20th day. We have isolated the samples in monsoon area. This is just a polishing. Means about that particular sample. What we have done
Okay.
basically, we have selected two plants, we have extracted their data in wet lab, and we have provided that data to the sequencing technologies, that is in Illumina, and they have generated data in the format of FASTA, and they given that data back to us, and we have analyzed the data below this step. This is the basically wet lab step. And next, now see,
But
before this, there is in the in second methodology section, maybe in the last paragraph, what they have done, yeah, in RNA transcription and transcriptomes after the sequencing, transcriptome assembly was carried before this. We have filtered the data. That filtered by the data by using some filter R tool. Yes, this is the tool by using which I have filtered the data. After that, I have assembled the data by using SOAP de novo, and then we have done the functional analysis. Functional analysis by meaning of BLASTX by using NR database. Is it visible?
yeah.
After that, what we have done, functional annotation and classification, where we have tried to find the pathways and some annotation at molecular, biological, and cellular level, where we have found some enzyme commission numbers, CAG pathways, then gene ontology annotations. Then I have written all these things in this place. Finally, what we have done, when we have found the genes which are overexpressing and down regulating, we have validated those genes. And after those genes, we have tried to find the protein-protein interaction network of those genes. This is the part that we have validated our data. After finding some key genes, we have validated the data and tried to map the data in the protein-protein interaction network. Finally, we have found which are the key genes which are responsible for the disease in a particular plant. Means in the same manner, you can design the experiment, means what you want to compare, either what you want to find from the particular organism. Okay. But I think this may be more difficult for you if we are going from one to another to hers. That's why today I want to just give a introduction. Then we'll download our data which is available in genome, because we can't sequence data right now. But what will we do? We'll download our data. Okay. The paper that I have recently published in TGPIN, I have just downloaded the data from NCBI SRA database. Yeah, you have seen the data. Okay. So this is the first step, how to download our data. In the second step, how to filter our data. In the third step, how to assemble the data. In the fourth step, how to find the functional annotation of the data, and then try to find the gene of interest, and then expression analysis of that particular gene, then validation of that particular gene. This is about NGS.
Yeah. So maybe we
Okay.
can take some sequence where the genome has not been established yet, or we
Yeah,
will be
when I have
taking.
done when I have done this work in 2016, the whole genome of T was not available. I have compared my genome sequence transcriptome sequences by using model plant Arabidopsis only,
So how do
because
you know that it is not available? Only NCBI you search and find out, or
no, there are some other tools as well. There is a tool, online genome database. There is another tool, genome in NCBI, Gold. There are some, and because if genome is available, there is a rush that genome of this particular plant is already available. If you will type, now there are two genomes are available. Fully genomes are not available. Only draft genomes of T are available. Draft means only partial sequences of those genes. Fully genomes are available, but their annotation is still not available in NCBI, because if anybody is going to do research on transcriptome or NGS, when I have done this research in 2016, I have submitted my data at NCBI. You can see in that paper, that same paper that you have opened, that data submitted at NCBI in SRA format by writing. Please open again. I'll show you and share the screen. Open the same paper.
Here it is NCBA.
There is a SRA as well. Yeah, see. Bioproject number sequence based SRA with a session number SRP 067 under bioproject this. From this bioproject, you can easily download the same data that I have submitted. Now, I have submitted this data for identification of disease resistant and disease susceptible genes. But maybe some other person, he will come and he just try to find the protein interaction network of some other 10 plants, and he wants to include this data as well. Then he can easily download this data and use this data. But there is a key. Key means when I have submitted this data in 2016, I have taken the extension that nobody can download this data up to 2 years. Up to 2 years, nobody can download my data. So that I will do everything possible with this data, and before any other person will download and analyze and publish his own paper without any spending a single penny. Because what we bioinformaticians can do, we can use the data. And what was the cost of the samples that we have done? You know, that was 20 lakh rupees for this sequencing project. And we have published one paper, and other person, a bioinformatician, will come, he will download the same data, he will find some new thing, and he will use that data, and he'll publish his own work in much more impact factor.
Okay. So then why did you deposit actually if you don't want anybody to see it for 2 years?
If I'll not deposit this data, who will trust on me that I have found that gene?
Okay, so you can tell that nobody should take it, and you
I
can still
told
deposit.
them nobody can download up to 2 years only, because
Okay.
I have to analyze this data up to basic level. But nobody has analyzed that. That is only a copyright for 2 years. Nobody has done anything with that data, and various other people, they have published their work by using the same data.
So
That's
this
why
20
bioinformatician is.
this
Yes.
20 lakh is for how many samples?
This data was, I think, it was for 16 samples were there, because we have done downloaded, we have
Of the
generated
leaf?
leaf conditions of from first day to 20th day, maybe first day, second, I think 14th day, 20th day, 7th day, and for each day we have sampled for both the plants, from infection to expression of the disease,
Okay.
for normal condition and disease condition.
So this was, I think, long back, right? Like when you were doing your PhD, maybe. I mean,
Yes,
five
that
at
was long
least
back.
10.
After that, I have again published another work with the same terms, that is in
Okay.
trillium govenium and stevia, and then apple. I have published the same things in the same, because when uh, I find the things that these things can be done by this uh, idea, then how to do? I have generated the data and submitted the data. After that, from the same data, I have published work in some other journals as well by using the same data. But in my next paper, what I have done, I have not said that we have generated the data. In my next paper, I have said that I have downloaded the data from NCBI. Because once I have submitted, that data belongs to NCBI only.
But if it is so costly, then how will people go for the sequencing?
Because, ma'am, I was working on CSIR, and in CSIR, they have their own Illumina, and they have all the chemicals available, and they have some huge projects related to tea board. Tea board have provided the fund of 5 crore rupees, and they need some sequencing because they want to get rid of this blister blight disease, due to which tea plant is continuously affecting. And in tea products there was a huge loss. And this
Mhm.
project was basically multinational project, because this project was done in the collaboration of China, this Sri Lanka, and our India. In Indian, my co-supervisor Dr. Ram Kumar Sharma was the member, and one TR Sharma, Dr. TR Sharma at IERI Delhi, he was the main coordinator of this project, because they
Okay.
want to know what kind of genes are there, they are affecting our tea plant, due to which basically maybe 1% or 2% economy is also affecting, because there is a huge import and export of tea.
Yeah, yeah. So
because
even now it is that much
because
costly, or the cost has reduced somehow?
ma'am, it depends, because how many samples you want to sequence. Because if you are generating the data your own, then it is a problem. But why we have to generate that data? There is a easy process, because we are bioinformaticians and we are doing some bioinformatics training as well. Just take data from experiment from other people, and analyze in the same manner and publish your work. But if you want to generate by your own, then it is costly, ma'am, because for each sample it may be 1 lakh rupees or 1.5 lakh rupees.
That's too much. Like a plant genome is small, right?
No,
So
ma'am, for
if they
t
go,
genome is 4 GB.
and what about a human genome? How big is
3
that compared
3
to
3
T?
GB, 3 GB.
So T genome is even more,
Even
4
more, even
GB.
more. Yes.
Oh,
So, you saw, like in the NCBI site, uh, like just now we saw that the data was a 5 GB, right?
No, no,
So,
that was the single sample.
right, right. So now when we download, so is it, I mean, possible that we can download that data and work on the laptop, or
Actually,
we need a
no, 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 no.
workstation?
It's very difficult to work on laptop. That's why I told you earlier. Laptop workstation is important for next generation sequencing analysis. That data is 5 GB size in compressed form. When you will extract the data from the compressed file, it will be 16 GB or 10 GB for the single sample. And if there are some 16 samples, just imagine how many what is the size of the whole data in raw format. Raw format is basically FASTQ format. That would be, I think, 100 GB or 200 GB for, if in average I'll imagine 8 GB per sample, for example. And if there are 16 samples, then you can see there may be some 100 to 130 GB data. And after filtering, the data will be 120 GB. But after assembly, the data will be reduced to MBs.
Okay.
Yeah. Then you will get the sequences, and you can easily find the functional annotation of sequences. And then you can find your gene of interest, and then expression analysis of that gene. And then you can do whatever you want to do, for example structural biology, protein protein interaction networks, or drug designing, whatever you want to do.
Then suppose someone is like doing some preliminary work on NGS and they don't have the workstation, then is it possible that
No, no,
they
no,
can
that is
do
not
some
possible, ma'am. That is the problem, that is the problem.
no, but
That
we
is
can
really
take
big.
no, we can take the bacteria which has less size, right? Probably 500 MB. The minimum
Basically,
size we can take, right? SRA, which has a minimum site, and try to do NGS.
but if size of the genome transcriptome is less and you want to do, there are 90% chances your laptop will crash, because I have lost 2 laptops. Then it was the beginning for me. For NGS, you need workstation, that is mandatory, because if 10,000 sequences are available, then the workstation should work for 24 into 7 in order to blast for each sequence one by one by one, one by one, one by one. If it will stop, then see how much time it will take. But if you keep on your laptop 24 into 7, then what will happen to our laptops? Surely it will crash.
So the ratio to the SRA size to the genome, the final genome is huge, is what you are saying, no?
No, no, no,
Only
no,
in
no. It
MBs you get
depends.
the genome.
MB's genome is not MB. This is transcriptome.
Okay.
Transcriptome means the data which is expressing in a particular condition. But genome
Okay.
means whatever the information is available. Means you don't have any particular target, just extract all the information. In human genome, they have not targeted the single gene or single thing. They just want to find how many genes are there and what is the size of each gene. They are not targeting the expression of that gene. They just want to know what is how much genes are available, what is the data of each gene, how many genes are there, how many proteins are there. That was their target. The genome is the separate thing. But for transcriptome, when we are working on transcriptome, for a particular condition, for a particular organism, you have to sequence. Okay.
Yeah, but transcriptome or genome, the methodology will be same. Sequencing methodology will
Methodology
be
is
same.
almost same.
Yeah. So
Methodology
then,
is
yeah.
almost same. Yeah. But for every genome or every transcriptome, 50% proteins or sequences are hypothetical. That was my main idea. If 50% sequences are hypothetical, then we are missing some important information. That's why I have published many papers in hypothetical side as well. Now, maybe you will be successful in that idea as well. Okay.
Oh, one question actually. What is the difference between a biosample that you showed in NCBI and the stain?
Basically, yeah. I'll show you. Biosample means when I have generated my data, there was a biosample. They have provided me how many samples you have, means how many biosamples you have. Then I have given that I have 16. They have given me ID of my biosample. Then they said, what is the project? I said, this is a Camellia sinensis. Then they said, biosamples, projects, and what is the ID? Means they are giving us some ID for a particular sample, for a particular project, because in NCBI there is a huge rush of this type of data. Then we have to assign the particular ID for each sample, for each project, for each organism, so that the data will be easily as available for other person who is trying to download that data. Biosample means that you have sequenced. Run means how many sequences you have done. For example, for first day there is a single run, for fourth day there is another run, for 15th day there is another run. These all things are basically related. I will tell you when we are going to download, because today I have shown you only a single sample, not even a full run, only a single sample. Okay. Yeah. These all terminologies are very good and very fine, because these are all for our convenience, so that we will be able to download the data in after knowing only ID of that sequence. Okay. No problem.
Yeah, so
Ma'am,
for this training, for this training, uh, those who are doing on laptop, how will they be able to do this?
they will be able to learn only. They will not able to, because first of all, what is their idea? What is their idea? If they are able to generate that idea, you can easily help them. And there are some space is also available at a computational center, IIT Delhi. They will give you access, and you can submit your data at that place, and you will analyze your data. But before that, your experiment should be very strong, so that you should proceed and do and write your paper.
Yeah, of course. I don't think so. They will give space for the training.
Because for
they
training they will not give space. This is just for learning, but for
correct.
doing a research, that is a separate thing.
Sure.
Because this is a learning part. Once you will learn, then you can manage your things in after joining a lab or after joining some research institute.
So maybe Partha would be knowing about the cloud computing. Is there any option?
Yeah, ma'am. Actually, that can be requested depending on what means like how much space we need. Uh, that is possible, but if we know what we are doing, we can actually get something from Google Cloud or Amazon Cloud.
But is that paid or how does that work?
I think I don't know, right? Amazon, and we need to check on that. I think for to some extent they will give a free subscription. That anyways you will get in our laptops. I think I'm not. I have to check on that. What instance they will give for free, and what instance they charge. Actually.
Okay, okay, okay. Yeah, just try to find some, even means how it is possible to find some space in order to do this type of work. Because I am here to teach you so that you can learn the things. Know how to implement these things, that is up to you only. Because whenever you will join a research lab, you can ask me, sir, I have done this, these are the results, what is the problem, what is the issue, I'll solve that. But if you will say I have to do, before that I need what kind of idea is there, why you are doing this, what is the importance, and all these things.
That is why I am.
So I'm sharing the genome of this zebrafish.
Yes,
So here, can you just comment on this? Like, what is the size of the data? Or
ma'am, just this is the SRS. You can easily, first of all, copy this run
It's 1.3. It's
SRR.
written on there. 1.3. It's
one,
written there.
yeah, 1.3 GB it's showing
Yeah,
Yes.
1.3 GB, this is compressed file.
size.
The file size basically it is 3.6 GB.
Okay.
Yeah,
What are the, like these designations?
ma'am, you are not audible. Please speak again.
I'm saying that there are several terms like hash of spots,
These are the
hash of.
spots. These are the bases. This is the size. This is the
Mm,
basis. Basis
Mhm.
basically ATGC. Spots means basically there. Whenever we are going to sequence, there is a ATGC like that. Means there is a. If you are aware of this Sanger sequencing, how many times that particular light flash was there in order to generate a particular image.
Okay.
mm. Maybe you can send this link, ma'am. Actually, I'll try to download and extract how much it will come to. We can see 1.3 GB
You
means
can
yeah,
just type this zebrafish,
yeah, yeah.
ZEBRA, zebrafish. So this is one experimental model, my favorite model.
Okay, 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 okay.
It's a fish
Ma'am,
actually,
just
yeah.
click on show abstract. Just click on show abstract.
Okay, just 1 minute. This one.
Yeah. So what they have reported, please see.
Overexpression of
See.
SOX3B
Yeah.
leading to
That is the
reduced
thing.
cytokine signaling.
That is the thing I want to convey from 1 hour. See, they have reported the overexpression of this gene. Same thing. Means they have tried to find a gene which was overexpressing and they have reported which was responsible for a particular thing. I don't know what was that. That is a cytokine signaling, but
Mhm.
no change in DNA methylation at this site. Overall design, neutrophils, and I means maybe you can understand these things because they are trying to find a particular gene which may have some effects.
Yeah, they are seeing the overexpression of this SOX3B.
Yeah, maybe this is a gene.
Yeah, yeah, this is a gene only.
Yes, yes. For identification of a particular gene in a particular condition, next generation sequencing is very important.
So
And
basically, all the
overexpression,
results Illumina are only giving, or what? Illumina. Mostly
Illumina,
I
there is
see.
a latest sequencing is Illumina HiSec,
This is Novasec.
Novasec, HiSec. There
Mm-hmm.
are some other varieties of Illumina because for each sequencing there is some upgradation. One can generate data in 2 days, other can generate data in 30 days.
Mm-hmm.
This is the only difference.
So all these are just a machines, is it? Illumina that you are saying is just a machine that will give you the
Yeah,
results,
Right.
yeah,
or
it's just a machine. It's just a machine.
And it takes several days, I think, to sequence the entire genome.
Yeah, it will take, ma'am, because for human genome project they have taken, I think, 10 years for human genome, and maybe for these particular things, there, yeah, time definitely we have to devote the time. I have devoted time of at least 4 years. After 4 years, I was able to learn what is NGS. Now I
Mm-hmm.
am conveying very, but not at the cost of 500, please.
Right.
That you discussed earlier. But yeah, you can easily see these type of things because